Hi guys, in this lecture we're going to cover the adrenergic transmission. So here and also we'll also cover the receptors. So why we need to understand this concept means here for the proper understanding of sympathetic drugs like sympathomimetics and sympatholytics. So we need to understand this concept. So here we already discussed the neurotransmitter in the adrenergic sympathetic nervous system, that is norepinephrine or noradrenaline. So that's why this system is we can also consider it as adrenergic system. And so as part of adrenergic transmission, here we study about the here adrenergic transmission is the story of five-step process. Here in this we'll cover the synthesis, storage, release, reuptake and pharmacological effects of norepinephrine, and also we'll cover the receptors for the norepinephrine. Okay, so here first we'll start with the adrenergic transmission. So here for the synthesis of norepinephrine, norepinephrine synthesis will begins with the uh, phenylalanine. Here for the means here the phenylalanine will acts as a precursor molecule for the synthesis of the norepinephrine. So here phenylalanine which is converted into the tyrosine in the liver, not within the nerve endings. So here the phenylalanine is converted into the tyrosine. So this tyrosine in the presence of phenylalanine hydroxylase. So this tyrosine is actively taken up into the nerve endings. So further process of noradrenaline will takes place within the nerve endings, adrenergic nerve endings. So here, so this tyrosine is taken up into the nerve endings by the active transporters. So upon taking tyrosine, so this tyrosine is converted into the dopa by the enzyme tyrosine hydroxylase. So this dopa is then converted into the dopamine by the dopa decarboxylase. So decarboxylation will takes place and will be converted into the dopamine. Okay. So and then after the formation of dopamine, so this dopamine is taken up into the vesicles, into the storage vesicles of this nerve endings. So within the storage vesicles only further process of noradrenaline synthesis will takes place. So here in these vesicles, this dopamine is converted into the noradrenaline in the presence of uh, beta hydroxylase enzyme. So this whatever the formed in our adrenaline, so which is stored within this storage vesicles until any stimulus comes for the release. Okay, so until here we discussed about the synthesis and storage. Stored noradrenaline is stored within the storage vesicles of the adrenergic nerve endings, right? Okay, so how it is released? So whenever any stimulus comes for the any stimulus comes towards the adrenergic system, so in the form of action potential. Okay, so here in response to this action potential, calcium will enters into the nerve endings. Okay, so calcium will enters into this nerve endings. So this calcium will causes the fusion of this vesicular membrane with this neuronal membrane. Okay, so fusion of this vesicular membrane with the neuronal membrane and causes the release of the noradrenaline into the nerve endings. Okay, so which is in response to the action potential and stimulus, nerve impulse. So this is called the nerve impulse coupled release. Okay, so then after the release, it will be this could be act on the receptors in order to show it effect. Okay, so the norepinephrine must be act on the receptors. So here for the noradrenaline, so receptors are the alpha and beta receptors, where these receptors are present either on the postganglionic neurons or all as well as in the effector organs. Okay, so whatever the released noradrenaline from postganglionic nerve endings will acts on the receptors present on the effector organs. So these receptors will be either alpha subtype or beta subtype. Okay, so once the norepinephrine will act on the receptors, okay, once the effect is produced, effect has to be terminated. Okay, so in order to stop the effect, so this the major mechanism in termination of this effect is this norepinephrine is again taken back into the nerve endings. Okay, so with the help of norepinephrine transporters here. Okay, so this norepinephrine is taken back into the, after showing the effect, it will be taken back into the nerve endings. Okay, so this will causes the termination of the effect. This is major mechanism for the termination of the effect of norepinephrine. Okay, so either it is metabolized by the monoamine oxidase and catechol-O-methyl transferase, okay, into the vinyl mandelic acid. Okay, so this is the another mechanism for the termination of the effect. Okay, so this is what the neurohumoral transmission of norepinephrine. Okay, so here some drugs will modify these mm, all these steps. Okay, some drugs will interfere with the synthesis, some will interfere with the storage, some will interfere with the release, and some will interfere with the reuptake, and some will interfere with the effects of norepinephrine on the receptors. Okay, receptor action, some will interfere with the receptor action, and some drugs will interfere with the metabolism. Let's see those drugs. Okay, so here in case of norepinephrine synthesis, conversion of tyrosine into the dopamine is the rate limiting step. Okay, so which is mediated through the tyrosine hydroxylase. Okay, so some of the drugs, so they acts by inhibiting this tyrosine hydroxylase, so will inhibits the availability of norepinephrine, inhibit the synthesis, right? So this can be mediated through me tyrosine. Okay, so me tyrosine is the drug, so that will interfere with the synthesis. How by inhibiting the tyrosine hydroxylase. So that enzyme is required for the conversion of tyrosine to the dopa. Okay, and then so again some other drugs. Okay, so that can also prevents the conversion of dopa to dopamine. Those are considered as dopa decarboxylase inhibitors, so such as carbidopa, benzorazide, etc. So that will interface with the, that also interface with the synthesis. Okay, so after the formation of dopamine or after the reuptake, so this norepinephrine must be taken up into the nerve endings for the storage until unless which is degraded by the MAO enzyme. Okay, so means which decreases the availability of norepinephrine. Okay, so that's why it must be taken up into the storage vesicles. Okay, so that this is mediated through this is considered as storage, right? So this storage is mediated through the active transporters. Those are vesicular monoamine transporters. Okay, vesicular monoamine transporters. So these vesicular monoamine transporters are inhibited by the drug that is reserpine, and nothing but here the synthesis is inhibited by me-tyrosine, so whereas storage is inhibited by the reserpine. Okay, so after that once the norepinephrine is stored, it does not directly released into the nerve endings, right? So for the release of noradrenaline into the nerve endings, it requires the nerve impulse. Okay, in the form of action potential. So this nerve impulse coupled release is inhibited by the guanethidine and bretylium. Guanethidine and bretylium. So these two drugs will interface with the release of norepinephrine. Okay, so then after this, so this norepinephrine after the release, it will act on the alpha and beta receptors. Okay, so the drugs here acts are considered as parasympathomimetics or parasympatholytics. Okay, adrenergic agonist or adrenergic antagonist. Okay, so and how the effect is terminated? So after showing the effect, so here effect is terminated by taking up the norepinephrine into back into the nerve endings, right? So this norepinephrine transport back into the nerve endings is mediated through the norepinephrine transporters. Okay, so this norepinephrine reuptake is inhibited by the some drugs. Okay, so those are considered as cocaine and tricyclic antidepressants like desipramine. Okay, so these drugs will inhibit the reuptake. Okay, so this is until the drugs that modify this adrenergic transmission. Okay, apart from this, some drugs here like tyramine and guanethidine, tyramine and ephedrine, etc. So here they interferes with the guanethidine displaces the noradrenaline from the storage vesicles into the cytoplasm. Okay, so whereas here the tyramine will act by exchange diffusion. Okay, so once the tyramine enters, norepinephrine is secreted. Okay, so one drug will exchange with the transport of other drug. Okay, so once the tyramine enters, norepinephrine will be released into the bloodstream. Okay, so this is all regarding the drugs that modifies the adrenergic transmission. Okay, so let's see the receptors and clear about the receptors. Okay, so here um, for showing the effect, norepinephrine must act on. Then we'll cover about the receptors, right? So what are the receptors for the norepinephrine? Here the receptors for the norepinephrine are alpha receptors and beta receptors. Okay, so these alpha receptors and beta receptors both are the G protein coupled receptors. So among that, this alpha 1 is the GQ subtype and the alpha 2 is the GA subtype. Whereas alpha 2 receptors are the GA subtype. So whereas beta 1, beta 2, beta 3 all are the GS subtype. Okay, so we'll clearly discuss what kind of receptors those are and what are the transducer mechanisms and where these receptors are present, and then so what are the pharmacological action, so what are the selective agonist and selective antagonist we'll discuss. Okay, so here coming to the next slide. So here first we'll start with the alpha receptors, right? So here alpha receptors are of how many types? So those are of two types. What are those? Alpha 1 and alpha 2. So this alpha 1 receptors are which type? GQ subtype, whereas alpha 2 receptors are the GA subtype. So what is the transducer mechanism for the GQ type? Whether it is alpha 1 or alpha 2, whatever the type, whatever the receptor it is. Okay, if it is GQ type, always the transducer mechanism is IP3 and DAG. Okay, and then here in case of GA type, always the transducer mechanism is decreased cyclic AMP. Okay, so then so these alpha 1 receptors are present on the blood vessels. So these are present on the blood vessels and also on the GAT and so GA sphincter. Okay, and last part sphincter, which is whose contraction is whose relaxation is responsible for defecation and whose contraction inhibits the defecation. Okay, and then also present on the bladder trigone muscles. Okay, so here sphincter muscles here also. Okay, sphincter muscles also contains alpha 1 receptors, and so the radial muscles of the eye. Okay, so radial muscles are nothing but dilator pupil which are associated with the pupil dilation, and also some male sexual organs which is associated with the ejaculation. Okay, so here coming to the functions, so here on blood vessels which is responsible for vasoconstriction, which causes the relaxation of entire GAT tract. Okay, alpha 1 receptors, but causes the contraction of the sphincter muscle. Okay, and then which contracts the trigone muscles. Okay, contracts the trigone muscles. So once the trigone muscle is compressed, okay, urination will be inhibited. Okay, so which causes the urinary retention. Okay, and also present on the radial muscles of the eye. Okay, so these radial muscle contraction is associated with the pupil dilation. Okay, and then uh, here seminal tract which is associated with the ejaculation. Okay, so all these are the functions of alpha 1 receptors. Whereas alpha 2 receptors, so these are the inhibitory type of receptors. So generally these are present on the preganglionic nerve endings itself. Okay, so from where it is released, the receptors are present within the nerve endings. This from the air the norepinephrine is released from. Okay, so this is associated with stimulation of this alpha 2 receptors are associated with decreased release of norepinephrine. Okay, so this is thing which is present on the adrenergic nerve terminals itself. Okay, and also it is present on the brain stem. Okay, in the medulla. Okay, stimulation of alpha 2 receptors in the medulla is decreases the adrenergic flow towards various organs. Adrenergic outflow towards the various organs means it decreases the adrenergic activity. Okay, and then again they are present on the blood vessels, and also present on the platelets, and also present on the beta cells of B cells of the pancreas. Okay, so here which is associated with platelet aggregation, so which is associated with the in decreased insulin secretion. Okay, so here coming to the agonist and antagonist. So here in case of alpha 1, so selective agonist is the phenylephrine and methoxamine. What do you mean by selective agonist? So they selectively stimulates the only alpha 1 receptors. It does not affect alpha 2, beta 1, beta 2 and beta 3. Okay, so those are phenylephrine and methoxamine. So what are the selective antagonists? Prazosin and terazosin. Okay, so here the selective agonist for the alpha 2 receptors are clonidine. Selective antagonists are ralphicin and yohimbine. Okay, so this is all regarding the alpha receptors. So then we'll continue with the beta receptors. So there are again three types of beta receptors, right? So what are the three types of there are three types of beta receptors, right? What are those? Beta 1, beta 2 and beta 3, right? So all these three are what? GS subtype. So in all these three, what is the transducer mechanism? Increased cyclic AMP. So whenever the GS subtype of receptors comes, so transducer mechanism is the cyclic AMP, increased cyclic AMP. Okay, so whereas beta 1, 1 and 1 present on the heart and also present on the juxtaglomerular cells of the kidney. Okay, so what are the functions? Which increases the heart rate, which increases the force of contraction, and which increases the AV conduction velocity. When this system is activated, so during fight and fright, right? So during emotion, stress, fear and all those conditions, this will be activated. Okay, so that's why it is associated with all these functions to increase the blood supply to various organs. Okay, and which is also present on the juxtaglomerular cells, which is associated with the renin release. Okay, so here beta 3 receptors, these are present on the adipose tissue and which is associated with the lipolysis. Okay, so here beta 2 receptors, those are present on all the smooth muscles and also on the skeletal muscles. Okay, here which is present on the bronchial smooth muscles associated with bronchodilation, and present on the where alpha 1 is present on the trigone muscle and associated with contraction, right? So here these are present on the detrusor muscles. So this detrusor muscle relaxation causes the holding of urine, right? Instead of urination, if it is compressed, so this will causes the urination, or if it is relaxed, so this will causes the urinary retention. Okay, they may cause the relaxation of the detrusor muscle and compression of the trigone muscle. But this trigone muscle receptor, on trigone muscle alpha 1 receptors are present, on detrusor muscle beta 2 receptors are present. Okay, and also on the GIT, which causes the relaxation of GIT and causes the decreases causes constipation. Okay, and here which is also present on the blood vessels associated with vasodilation. Okay, and also present on the uterus, causes uterine relaxation. So which is present on the liver, which is associated with glycogenolysis and increased glucose concentration. Okay, and then on eye. Okay, so which is not the prominent effect, but which is also present on the eye and are associated with the ciliary muscle relaxation. Okay, so these all are the functions of the beta 1, beta 2 and beta 3 receptors. Okay, so if you understand this, you can clearly understand the sympathetic drugs. Now you know, so what are the adrenergic distribution, which is distributed to the heart, kidney and then bronchial smooth muscle, bladder, blood vessels, uterus, eye, adipose tissue, platelets, and you know distributed organs, and you also know what are the functions of those organs, adrenergic actions, and you also know how these functions are produced with the help of receptors. When the norepinephrine acts on these receptors only, all these effects will be produced. Okay, so then you can clearly understand the sympathomimetics and sympathomimesis. lytics. Okay, I hope you understand this class. Okay, if you like the class,